My name is Sagiv Shiffman. I'm from the Hebrew University of Jerusalem. I really want to thank the organizers for feeding us all the time and changing significantly my microbiome. And so I'm actually a biologist, a geneticist. We do experiments in my lab. Most of the work is experimental. And I'm going to talk today, I'll give you some overview about what I think about mainly autism and the involvement of rare and de novo mutations in autism and other neurodevelopmental disorders. Okay, so this is the topic. Okay, so I want to start by telling you why do I study neurodevelopmental disorders. It's kind of a selfish reason. I'm interested in social science, in social behavior. More generally, I'm interested in what I call BB. It's brain and behavior. But I'm not sure that this is what the public really wants. I think that the public wants different things. They want things like treatment, rehabilitation, medicine, and prevention. So very different things. And so as a geneticist, we always promise that our research will benefit the public. So the promise of genetics include the fact that if we identify the genetic risk variants, we will be able to predict the diseases in a better way. And this is very important, especially for disorders like autism. We know that early intervention really helps. The other thing that we tell everybody is that if we identify the genetic risk variants, we will understand the biology and we will be able to develop treatments, better treatments based on the biological knowledge. But I think, so we have very high expectations, but I think we need also to face reality. Okay, so I'm going to talk quite a lot about problems and disappointments. Okay, so we need to think about the expectations versus the reality. In our expectations that every time we have a new technology, we think that there will be a huge progress, we will solve all the problems. We had it with the microarrays when they were introduced, and then with the new sequencing technologies. But in fact, when we face reality, we have disappointment. So we had disappointment from genome-wide association studies looking at common variations. We have disappointment also from the progress that we are making with sequencing genomes and looking for rare variations. So I want to start with just giving you an example of expectations versus reality. Okay? So before genome-wide association studies, Francis Collins, this is from 2006, he was interviewed to the Wall Street Journal. This was just before when the 500,000 SNP arrays was introduced, and he said, I expect that there are about 12 genes involved. He was talking about diabetes, type 2 diabetes, and that all of them will be discovered in the next 2 years. Think about it, with 12 genes, we can predict diabetes, we'll have a specific pathway, and we will develop treatments based on those. 10 years later, of course, we need to face reality. We don't talk about polygenic inheritance anymore. As probably many of you know, this paper that was published by Jonathan Pritchard in Cell in 2017, we're talking about an omnigenic model. And this is from the paper. I'll read it. What is this omnigenic? Many complex traits are driven by enormously large number of variants of small effects, potentially implicating most regulatory variants that are active in the disease-relevant tissues. So instead of talking about 12 genes, we're talking about now that the whole genome is actually associated with a disease or with a trait. All regulatory variants that will be active in the right tissue, for example, if we're talking about schizophrenia, any gene that is expressed in the brain will be associated with schizophrenia. So from a specific pathway, we are having the whole genome associated with the trait. So this is a bit of a disappointment, both for predicting and for developing new treatments. But in the same paper, we have already a solution. Okay? So the solution that they propose is that the next most promising step is to hunt for lower frequency variants of larger effects. Okay? So we have the new expectations, and I want you to face reality and share with you the next disappointment. Okay? So for rare variants, we've been studying rare variants for more than 10 years in autism and other neurodevelopmental disorders, and so we can face the disappointment already now. So with autism, there are different types of variants that are associated with autism, from de novo mutations, rare mutations, and common variants. All of them contribute to autism. When I'm talking about de novo mutations, we're talking about a variant that we see in a child with autism, and we don't see this variant in the parents. Okay, so this is a de novo mutation. Normally, this variant will be transmitted from the father's side. We also know that there are rare inherited variants that contribute to autism. We also talk about ultra rare variants that we see in a specific family, but we don't see in any other controls that we look at. And of course, there are the common variants, all those alleles that are everywhere in all populations, and the accumulations of many risk variants, both from the father and from the mother, together increases the risk of having a neurodevelopmental disorder like autism. Now, but almost all the information that we have now on autism actually comes from de novo mutations, what I call here the rarest of them all. Okay, so those mutations, of course, because they are rare, have much larger effect sizes, and we have something like 100 genes that are being now implicated in autism, all of them because of de novo mutations. Now, I want to share with you another disappointment. I want to disappoint you and tell you that we all have de novo mutations, all of you. Yes. On average, each one of you has something like 60 de novo mutations in your genome. Okay? And think about it. Think about a child that is just born, and he has these 60 mutations. They are distributed relatively uniform in the genome. And so if you're unlucky, you get this de novo mutation in a critical gene, and you are likely to develop some disorder. So as a person coming from Israel, from the Middle East, this is for me, sounds like war. Yeah? Somebody is shooting at you, those mutations. And reminds me some statistical study that was done in World War II by Abraham Wald. He was studying the distribution of damage to aircraft and was trying to improve the aircraft against bullets. So he was a statistician, Abraham Wald, and he proposed that the bullets, the hits, are uniformly distributed in the airplanes, and by studying those airplanes, they did come back to the airfield, we can recognize those regions that are missing bullets. Those should be the critical regions of the plane that we need to protect. So we need to actually to protect those places where we don't see bullets, because we need to remember that there are many airplanes that didn't come back because they were hit in those critical regions. So our genome is like an aircraft in a sense, that we can learn from looking at mutations in the genome, both about neurodevelopmental disorders. We can look and see what kind of genes are being hit by de novo mutations in individuals with neurodevelopmental disorders. But we can learn quite a lot from looking at normally developing individuals and see what type of genes have lower frequency of mutation, lower than what you would expect by chance. And those genes, we call them also essential genes. As I said, we can learn what type of processes are sensitive to specific type of mutations. So for de novo mutations in autism, it's all started from a new technology. The new technology was, in the past, was microarrays. For the first time, we could identify deletions, duplications, which are small, that we cannot see under the microscope. And with this technology, in 2007, people showed that de novo copy number variations are much more common in patients. This is a paper published in Science. And they've just compared frequencies of de novo copy number variations between patients and controls. And these are the numbers. This is a study from the Wigler's group. So you see here the numbers. Around 1% of control individuals have those de novo copy number variations, relative to what we call simplex families, that they have 7 to 10% of those copy number variations. Simplex families, or sporadic cases, are families when you have just one individual with autism. Okay? If you look at multiplex families or familial cases where you have multiple kids with autism, you don't see a significant enrichment of those copy number variations. And this is logical, because if you have a familial cases, you will think that there is an inherited risk factors in these families. But this was not enough. Michael Wigler and his group claimed that actually de novo mutation can explain also the familial cases. Okay? So in a paper in PNAS, they've suggested that autism may be the result of dominantly acting de novo mutations that have reduced penetrance in females, and this could explain both familial or multiplex families and also simplex families. And the way it goes is like this. You have the de novo mutation. If it happens in a boy, it will be autistic, and then normally will not have kids, and this is a dead end of this mutation in the population. However, if this de novo mutation will happen in a girl, because of the low penetrance, specifically in females, there is some likelihood that she will marry or have kids without marrying, but this will create a high-risk family. She will transmit this mutation to half of her kids, and if there are boys, they were highly likely to be autistic. So this was their model, and you're not going to be surprised that the focus has shifted towards looking at rare variants in autism. And here what you see is a cover of Neuron from 2011. There were several publications about copy number variations in autism. Okay? High expectations, but now let's face reality. Okay? So I've opened the paper and I listed all the genes that were affected by de novo copy number variations just from one of those papers in this journal. And here they are. Okay? Quite a lot. I mean, I can send you the list of genes and you can look and see if you can make sense out of it, but it's very, very hard. And so we have several problems here. One is that each CNV contains many, many different genes, and it's very hard to distinguish between what I call here drivers and passengers, the really causative genes, and the ones that are just close to this risk genes. Okay, so this is one problem that we're struggling to solve. Another problem is that if you look at different individuals, each individual has a very different CNV. Most of them are not recurrent. And so we have so many genes, and one of the questions is, what are the shared processes that are affected by multiple autism-associated genes? Okay, so this is two questions that we've been trying to solve. With this kind of problems, it's very good that there are new technologies with we have new expectations that maybe they can help us solve those problems. And the new technology is that now it's not new technology anymore, is exome sequencing. You can, using exome sequencing, you can enrich your sample just for the coding regions of the genome, sequence them, and identify de novo single nucleotide variations. So these are just point mutations that we can identify. And what's good about point mutations is that it's very clear which gene is affected, which gene is important, unlike the copy number variations. And we can identify very different type of mutations. If this is the wild type, the protein sequence of the wild type RNA and DNA, we can identify mutations that we call loss of function mutations or disruptive mutations that are highly likely to be functional, very easy to predict those mutations. And here in this example, we have a mutation that causes a stop codon, which will truncate the protein. We also see missense mutations. The cases where here, the example here is that the proline amino acid is changed to another glutamate, another amino acid. Those mutations could be functional, but it's relatively hard to predict which one will be functional, which one is not. Okay. And we have those mutations that have an effect on DNA or RNA sequence, but don't change the protein level, and probably most of them are not functional, and that's why we call them silent mutations, or more officially synonymous mutations. And very similar to the copy number variations, we see a higher frequency of those type of functional mutations, de novo functional point mutations in autism. This has been shown by many different studies. Here is just one of them. What you see here is a family, and we have the autistic kid, and there is an estimation of the average odds ratio, the risk of having in gray a de novo point mutation, which is a disruptive, a de novo CNV. You can see that there is a significantly higher risk relative to inherited copy number variation, which has a very modest effect. In this plot, you can also see that in most cases, those mutations are coming from the father to the child. So we have those point mutations, and this should be very helpful also maybe to study the copy number variations. I mean, we can have the expectation that the de novo point mutations of SNVs can be used to distinguish between the drivers and passengers in copy number variations. Makes sense, no? But you see that I wrote your expectations, so you know I'm going to show you the reality now, and this is not really in most cases, because when we look at large de novo copy number variations and de novo point mutations, they target different genes most of the time. Okay, and I'll just show you the data for that. What you see here are genes that are here inside de novo copy number variations. These are genes not in copy number variations, and the different colors here are for amounts of de novo loss of function, de novo missense, and de novo silent mutations that we see in them. And you can see that there is no difference between genes in CNVs and genes not in CNVs. Okay, no enrichment. If we take those genes in CNV, de novo CNVs, and start dividing them into different categories, we do see an enrichment or increase in the amount of de novo loss of function mutation and missense mutation only in the ones which are small deletions with not many genes in them. But most of the copy number variations in autism are actually large here. So this was the small, this is the large deletions with many, many genes. And here you see that there is no increase in the amount of loss of function mutations or missense mutations. What does it mean? It means that those large de novo copy number variations probably contain multiple risk genes that interact together additively or epistatically. We don't really know. Okay, so this is what it means. The other question I was asking is, what are the shared processes that are affected by multiple autism-associated genes? There have been multiple type of methods and strategies to identify shared processes and take those genes and trying to understand what type of brain regions or developmental stages are involved in the disorder. So, for example, one approach is based on the assumption that functionally related genes will be co-expressed together and they will be co-expressed in a specific brain region or cells or time point, which is relevant to the disorder. So people have been using, including ourselves, in approaches like weighted gene co-expression network analysis to try and find those co-expressed genes and then link them to specific brain regions. So this is one approach. Another approach has been to look whether the risk genes are preferentially expressed in specific brain regions, cells, time points, etc., which are relevant to this disorder. Preferentially expressed meaning that we are looking for those, for example, brain regions that show high expression of those genes more than other regions in the brain. So there have been multiple methods to find those preferentially expressed regions. This is one such a method, the tissue-specific expression analysis. More simple approaches are just to take all those risk genes and look for enrichment for pathways or biological processes. And the promise is that you take all those risk genes and you're going to find processes or pathways that are important for the disorder. So this is the expectations. In reality, what we see is that the processes or the brain regions, etc., are not specific to the disorder. They're actually telling us something about the type of mutations that we're studying. So they're specific to the mutations. So instead of finding the pathways that are associated with the disorder, we're finding a pathway that is just sensitive to a specific type of mutations. And just to summarize it, I'm not going to show you all the data, but when we compare loss of function mutations, missense mutations, and recessive mutations, we find very different things, very different pathways. So for example, for loss of function mutations, we find that those genes are enriched for chromatin and transcription regulations that are involved in the proliferation and differentiation of neurons. So very early stages of brain development. If you look at missense mutations, you're going to find different pathways. For example, pathways like neuron development later in brain development. And if you look at, and those both of them are dominant mutations. We're talking about de novo mutations. One copy is being affected. If we look at recessive mutations that are associated with neurodevelopmental disorders, we see the very different pathways. Something like things like metabolism. So you have enzymes that are affected by recessive mutations because they're sensitive to this type of mutations, and enzymes are not sensitive to mutations in one copy. And we think we understand why those type of genes are more sensitive to loss of function mutation, why those type of genes are more sensitive to missense mutations. So when you look at the chromatin and transcription regulation, you see that they are very tightly regulated, their expression level. So for example, if we look at single cells, we see a very low expression variation for those type of genes. For the missense mutations, we see that there are, if you look at a protein-protein interaction network, there are kind of hub proteins. So you have a lot of interactions with other proteins. And in fact, there was just publication now in Nature Genetics, a method that is used to predict functional missense mutations based on whether the mutation is likely to affect protein-protein interactions. So this is in the level of the group of genes, okay? Of course, we want also to identify specific genes, which genes are associated with the disease, so we do it just in a type of an association study with rare variants. And there are all kinds of problems with this approach. It's been solved by different methods. One problem is, of course, as I told you, we all have de novo mutations, so you need to have some kind of a control. It's not enough just to say, I see a lot of de novo mutations in this particular gene in autistic individuals. But many of the studies lack control individuals because they've collected trios, just 2 parents and a child. And even those studies that do have control individuals, there is quite limited information because the number of functional de novo mutations in each of the genome is very small, maybe 1 or 2, so not a lot of information. So there have been several solutions in the literature. One of them is just to simulate random mutations. This is, for example, the DNReach software. So when you account for the gene size, the sequence context, and functional effect of mutations, you can just randomly generate mutations and use it to test your hypothesis. So, for example, we've used this software to test whether there is a significant overlap between disorders. Again, you cannot just see that there is a gene that is mutated in both schizophrenia and autism and say there is an overlap, because some of those genes have mutations just because, for example, they are very, very large. Okay? So using this software, we could show, and others showed it before also, that there is a very, very significant overlap between different disorders. So this is part of the low specificity that we have for de novo mutations. Here are the results for loss of function mutations, missense mutations, and synonymous mutations. And I'm comparing here intellectual disability with autism, intellectual disability with schizophrenia, and autism with schizophrenia. And here you have the observed versus the expected ratio. You can see that we have a significant overlap both for loss of function mutations and for missense mutations. Much stronger signals when we compare intellectual disability to each one of those disorders, and we don't see any overlap with the synonymous silent mutations that are treated as a control. So this is just one approach. Another approach is based on normally developing individuals. You can use a similar model and go over every gene in the genome and ask whether the number of mutations that you're observed are higher, equal, or lower than what you would expect by chance. And by doing it, you can identify those genes that lack, that are constrained or are intolerant to mutations. This also can help you to identify those genes that are associated with the disorders. There have been several methods to do it. One of them was published in 2014 by Mike Daly and his group in Nature Genetics. In this paper, they use this framework to assign for each gene in the genome a Z-score which contains the amounts of mutations that you observe versus what you would expect by chance. And if you want, you can put a cutoff here and say those are this is the genes that are most intolerant to mutations in the genome. Okay? But for each gene, you have a quantitative measure of how intolerant to mutation it is. And if you look at those genes that are the most intolerant to mutations, first of all, we see that they are involved in the brain. So, for example, if we look if they are preferentially expressed in any tissue in our body, we see that they are most enriched in the brain. Here I'm just using this the tissue-specific expression analysis tool from the Garty lab. You can just put the gene list And I'm not going to go into the details, but what we're testing here is whether there is a enrichment in preferential expression in any tissue. And the red here indicates that there is those genes are preferentially expressed in the brain and not in other tissues. Not surprisingly, we also see an overlap between those genes that are most intolerant to mutations and genes that are being mutated in neurodevelopmental disorders across different neurodevelopmental disorders. Again, this is something that was shown also by other groups. I'm just going to show you the results. Here what we are doing is looking at the mean of the Z-score for genes that are with different type of mutations in loss of function, missense, and synonymous mutations in different disorders. Again, intellectual disability, autism, schizophrenia, and control. And you can see that both for loss of function mutations and missense mutations, we see a higher Z-score relative to the control, highest for intellectual disability, both for loss of function mutations and for missense mutations, but we don't see any effect for synonymous mutations, again, that there are used as a type of a control. I want now to move to a new technology that we are using in my lab, an experimental new technology, give you some expectations, and we'll need to wait a few years to get the disappointment probably. But currently we're excited, okay? It's funny for you, but I'm crying inside. Okay, so the experimental approach, so all those microarrays and sequencing are new technologies to read the genome, but we have also new technologies to manipulate or to write the genome, okay? To manipulate the genome. And one of them are of course, you've all heard about CRISPR, I'm sure, and but you can use CRISPR not just to point to manipulate one specific gene, you can do it across the genome, okay? So the way we're doing it now is using a CRISPR-Cas9 approach to manipulate all genes in the genome, okay? And then you can manipulate all the genes in the genome and take your cells and do all kind of things with them and study, for example, in this scenario here, we're studying what are the genes that are important for different stages of neuronal differentiation. And the way it goes is that first you take, in our case, what we use is mouse embryonic stem cells, transfect them with a Cas9, first of all, and then you transfect them with a pooled library of guide RNAs. So you take guide RNAs that target all the genes in the genome in one pool, they are in one tube, and you can transfect all the cells with a pool of those guide RNAs, and you do it in a way that each cell will receive just one guide RNA. So in each cell in the culture, each cell will have a mutation in a single gene, okay? So this is the CRISPR library, you transfect the mouse embryonic stem cells. Now you can do all kind of things, you can just grow the mouse embryonic stem cells and observe what's going on, or you can differentiate the cells into neural progenitor cells and neurons. And we can monitor what's going on here by just sequencing the guide RNAs. So we are sequencing the guide RNAs and looking at the frequency of the guide RNAs, and we can identify those guide RNAs that are negatively selected, those kind of guide RNAs that will go out of the population. Those guide RNAs that are positively selected are the ones that we will see increase in their frequency. And so in a sense, what happens in this dish is like a rapid evolution. So because we're generating mutations in all the genes, we have negative and positive selection. And unfortunately, we have also genetic drift because the number of cells that have each one of those guide RNAs are quite limited, several hundreds. So we have also random changes in the frequency of those guide RNAs each time that the cells are dividing, okay? And I'll just start and describe just a little bit about what we did with mouse embryonic stem cells before differentiation. So the pool that we are using, the CRISPR pool library that we're using, target almost 20,000 genes in the mouse genome. Each gene has 4 guide RNAs. We also, something very important for us is that we have 1,000 control guide RNAs. Those are guide RNAs that don't target any gene in the mouse genome. And what we did was to sequence those guide RNAs in different intervals after 8, 11, 15, and 18 days in vitro. So we just give the mouse embryonic stem cells the opportunity to proliferate, okay, in this experiment. We don't do anything else. So the next step is to find those guide RNAs that change significantly with time, okay? The way we do it, not sure that this is the best way, you should tell me, but we're using simulation with the control guide RNAs to establish significance. What we're doing is, as I said, for each gene we have 4 guide RNAs. In each day we rank all the guide RNAs based on their fold change. So we're going to have from 1 to almost 80,000 guide RNAs. And then for each gene, so you're going to have a rank for each day, and we just look at the sum of all the ranks across all days. And we do the same thing in a simulation by taking randomly 4 guide RNAs from the control guide RNAs. And we compare the distributions. This is what you see here. In red, this is the distribution of the control guide RNAs, and in blue, it's the guide RNAs that target genes. And based on that, we can assign significance for changes in both directions, positive and negative. Okay. For us, it looks like it works very well because it's highly reproducible between screens. Here, what you see as comparing our results to a screen that was done in very different cells, different statistical method, different conditions, and different CRISPR pooled library. And so in red are the genes that were non-significant in the previous screen. In blue are the ones that were significant. And here you see the fold change, the density plot of the fold change in our screen. And you can see that there is a big difference. And also if you do the overlap between the significant genes in our study and their study, it's highly significant. Something like 64% are shared. So using this screen, we're able to, as I said, to find negatively and positively selected genes. Most of the genes that we see are actually negatively selected. In this volcano plot here, you have the p-value in a log 10 scale and the fold change in day 18. The line here is just the significant threshold. And we find 2,379 negatively selected genes and 38 positively selected genes. And if you're going to look at this list of genes that are very non-random, we have entire pathways that are being negatively selected. Also, the positively selected genes are genes, for example, genes that are known to be involved in cancer. If we compare it to other data sets like the constrained genes in humans that I've told you about, we see a very significant overlap, but you can see that it's not complete for many different reasons, but we can discuss later if you want. We also see that the genes that are essential in mouse embryonic stem cells are specifically associated with neurodevelopmental disorders, although we were just looking at mouse embryonic stem cells before differentiation. So if we compare different individuals with developmental delay, so there are individuals with developmental disorders which affect the brain, this is NDDs here, and individuals with developmental disorders that do not affect the brain, and we look at their de novo mutations and compare those genes with the genes that are essential in mouse embryonic stem cells, and we see a strong enrichment only for the ones that have disorders with phenotypes that are related to the brain. 2 minutes, okay. I'm going to talk another one, okay? The other thing, I told you that many of the methods that we've been using to try and identify functionally related genes or to identify the brain region which is relevant to the disorder are based on gene expression. So our assumption is that if the genes are expressed in a specific or preferentially expressed in a specific tissue or time, this will be the relevant tissue or relevant time to the disease. So we can do the same thing here for this experimental study and see whether genes that are expressed, for example, in mouse embryonic stem cells, are the ones that are more essential. So in general, the answer is yes. Yeah. So here, what you see, I'll go over it briefly, is we have a heat map of gene expression of many different genes. All of them are related to neurodevelopmental disorders, and we have a differentiation day. This is during taking human embryonic stem cell, differentiating them into cortical neurons, and you have different days here. And we've clustered those genes into 4 groups. The group here is the group that are mostly expressed in embryonic stem cells, and this last group, for example, are the those genes are expressed more in neurons. And if you look at the sum of ranks that I've told you about in our screen, you can see that there is this shift in the distribution, but also you have here the number of genes in each one of those 4 groups that are essential, and you can see that we have the highest number of essential genes for genes that are expressed in mouse embryonic stem cells. Okay, you can also see it here. I don't know if you can see it in these red lines, which indicate where are the essential genes located. So just to summarize what I was trying to say about the disappointment that we have from the low specificity of de novo mutations, that is resemble what we see from genome-wide association studies. So analyzing de novo mutations, we reveal the processes that are sensitive to the type of mutations and not necessarily processes that are specific to the disease. I told you that we also see a very strong genetic overlap between different neurodevelopmental disorders. You can use the distribution of de novo mutations in the genome to study the critical genes and pathways that are sensitive, but they are sensitive to the specific type of mutation, in mutations in one copy. I told you that neurodevelopmental genes, the ones that are implicated by de novo single nucleotide variants, are actually essential genes. They are just sensitive to mutations in one copy, or you can call them haploinsufficient genes. And the fact that we have lack of specificity is a major challenge for what I call Trump, for the developing treatments and medicine, etc. So I want to thank people in my lab. This work that I've presented, most of it was done by Shachar Shochat. She was here in this course next year. She's been doing both the statistical analysis and the experimental work. And thank you for your attention. I don't have time for questions, or?
Oh, I was going to ask questions. I think everybody wants coffee.
Yes.
So,
I'll ask myself some questions in the break.
I have one. I didn't really catch what was your sorting criteria for this CRISPR screen. Like, what was the plus and minus?
The plus and minus. In the CRISPR screen, which one? Here? Here?
When you introduced that, right? Like, so, yeah, so what is your selection? So what does positive mean, for example?
Positive selection means specific guide RNAs that will increase in frequency during the process that we do. So if you're just proliferating the cells, you will see that there are specific guide RNAs that will increase. So they give advantage to the cells. So these guide RNAs will mutate specific genes that, for example, will increase the proliferation rate of the cells. So you will see those guide RNAs increasing with time. So this type of genes are, for example, genes that are related to cancer.
Okay. So you're comparing like day 8 versus day 11?
Yes. Or from the first the representation of those guide RNAs in the beginning. Okay, sorry that it was not clear.
Questions?
Thanks.
Let's go ahead and take the speaker again.